is my pleasure to introduce Dr. Paul O'Shea. Dr. Paul O'Shea is a senior R&D leader with a doctorate and 25 years of scientific and operational leadership, a forward-looking and progressive R&D leader inspiring the creation of 5 new scientific groups. Paul has a PhD in biochemistry from the University of Ireland in Galway. His company, Exemplify Biopharma, provides integrated end-to-end program and or portfolio-based CMC drug development deliverables to pharmaceutical and biotech partners. Today, he'll be talking to us about the role of CMC in IND Enabling Studies.
Thank you very much for the introduction. Let me just share my screen and okay. It's all visible now? We're all good?
Yes, all.
Okay. All right. Thank you. Okay, so let me get started. So today I'm going to talk a little bit about CMC. So in a general sense, what my outline is, what is CMC for those who are not familiar with it. And specifically, I'm going to talk about the IND Enabling CMC activities, which are a very important set of activities that are essentially helping move molecules from, let's say, the world of preclinical into the clinic. And I'll talk about some of the technical deliverables from a CMC perspective, and also it's obviously a regulated environment, so what are the regulatory deliverables? And then a little bit about the transition, because there is a transition on many levels from the discovery environment, let's say, into the development environment. And at the end, maybe I'll talk just briefly about my company, Exemplify Biopharma. So what is CMC? So CMC has a number of different functional areas essentially associated with it. There are, 5 functional areas: process chemistry, analytical chemistry, formulation development, quality assurance, and regulatory affairs. And all the activities of CMC pretty much fall under these, let's say, functional areas. So CMC is a term that's come from the FDA essentially, and it's an acronym that stands for Chemistry, Manufacturing, and Controls. And essentially, it's coming from a section of your IND or NDA, let's say, submission, and that's Module 3. So all of the information concerning chemistry, manufacturing, controls is essentially contained in Module 3 of your IND submission. So what exactly is the agency looking for when you submit an IND, and what are they looking for from the CMC component? Essentially, they're looking for information about the manufacturer. What's the drug? What's its composition? Is it stable? And also, what controls are used when you're making the drug substance or the drug product? And all that information needs to be provided to the agency as part of your submission. Why are they asking for this? Essentially, it's about confidence. It's about saying that you know what you're doing, you know how to do it, and you're not going to jeopardize in any way, in any shape or form, patient safety, so that you can consistently make material, you can consistently analyze it, you can get consistent results, and all of that pointing towards confidence that it's suitable to dose to a human. So what are the technical deliverables? From the CMC perspective, you need a formulation to support your toxicology and your pharmacology studies. Sometimes this is very straightforward, other times it can be very challenging. You need a scalable synthesis for your drug substance, because you're going to have to deliver, obviously, drug substance to support everything that's happening in the clinical arena, not just supplying the clinic, but all of your toxicology studies, your pharmacology studies, all of your preclinical studies will need drug to support them. You also need an appropriate phase for your drug substance. So will it be the free form? Should it be a salt, a polymorph, or what exactly is the form of that final API going to be? You'll need to be able to formulate it, and you'll need to have a scalable formulation, because you're going to have to manufacture your phase 1 clinical supplies. So whether that's a capsule, tablet, or even an on-site formulation, you still need to establish the stability of that material. You also need obviously all of the appropriate analytical methods. So you need to be able to characterize your drug substance and drug product, report the assay, the purity, the impurities, and all of that type of information. And you also need stability data to support your drug substance and drug product. Typically, that stability data is enabling you to run the study because you want to be able to show the agency, for example, that during the course of your phase 1 study, your drug product, your formulation won't go bad, essentially, or won't go off. And you need to characterize the molecule itself. So, what's the structure, proof of structure, and things like that. So they're all the sort of technical things that the agency expects to see and that, are done as part of this whole workup, let's say, to get into the clinic. In terms of the regulatory deliverables themselves, as I mentioned, it's Module 3 of the IND filing. And what are the sort of requirements? Because this is obviously a regulated document in a sense for a regulated industry. So you have to provide certain information as part of your IND filing. So you need to know what the molecular properties are. You need to disclose that and how you're characterizing it. So what's the structure? What does it physically behave? What does it look like essentially? You have to disclose all of your manufacturing and your analytical methods. So you have to provide a process description, the methods you're using to release the material, what your specifications are, and also the batch history. So what's been the purity and so forth for previous batches that you've made to support your entry into the clinic. You need the equivalent information for the formulation, including any information about a placebo if you're planning to run a placebo-controlled study in phase 1. You need to know the packaging configuration. So is your drug substance in a bottle? Is it in a plastic bag? Is it in what kind of container is it in? And the same for the formulation. Is it capsules in a bottle? Is it a blister pack? So you have to provide all this information. And finally, the stability. So you need to show that your drug substance and your drug product are essentially stable, at least for the duration of the studies that you're going to run. So all of these activities that I've outlined are all sort of in the CMC bucket, but they all need to happen to enable you to get into the clinic, provided your toxicology data and so forth are appropriate. So in a transitional sense, the project is essentially moving from what I would describe as a discovery environment into a development environment, and actually these are quite different. I have just some simple definitions, so if you're a medicinal chemist, or don't take offense at my simplistic definition, but in a sense, the discovery process, if you want to call it that, is pretty much an iterative process. You're trying to refine a structure or a core molecule with the right features to get the best overall balance of properties. And those properties are obviously things like potency, selectivity, and so forth, but there are a lot of properties that need to be considered to have a successful drug molecule. In terms of preclinical development, again, this is a pretty simplistic definition, but it's essentially all the tasks that you need to have are in place, and all the data you need in place to advance your selected drug candidate through manufacturing, formulation, the pharmacology, pharmacokinetic, and toxicology testing. So in other words, you need to show to the agency through all of these different functions, and there are other functions, that your molecule is suitable, has suitable properties to enable it to go into the clinic. In terms of the next stage of development, which is really clinical development, the focus is actually quite different. So now you have only one molecule, and you need to understand all the properties of that molecule. You need to understand specifically how to make that molecule and the clinical performance of that single molecule. So you're sort of going way down into the weeds about this particular molecule and all its properties, all its behaviors, all its good points and, let's say, bad points, depending on exactly what you're studying it for. So in essence, you have to select a molecule, and part of that selection process is certainly a term we use, which is molecule developability. So how do you really figure out whether your molecule has the properties and the legs, let's say, to go all the way to the marketplace if it's successful? So essentially, this is looking at all the intrinsic properties of the specific molecule. It really should be done as part of the preclinical package, because in essence, you really want to have confidence in the molecule that as I say, if your clinical data are good, that this molecule can actually be developed. It's typically, I would say, requires information and input from a number of different functional areas because, again, as I mentioned earlier, you're talking about balancing the properties of a particular molecule and trying to get the right balance. So obviously there's a very strong input from the medicinal chemistry folks. You need to understand the pharmacology of the molecule, how does it behave. You need to also understand, obviously, the toxicology and the limitations on toxicology, all the ADME properties from a drug metabolism point of view. And, that sort of, let's call it poor relation until recently, right down at the bottom here is CMC. So in the past, I would say a lot of companies had no input from CMC in terms of molecule developability, which, at least in my opinion, is a little bit short-sighted because a lot of molecules in the past have dropped out of development because they have very poor CMC properties, meaning they're either very insoluble or you can't formulate them, or in some cases you can't really even make them on scale. So in recent times, meaning probably the last 5 to 10 years, I think the CMC aspect of this whole getting into the clinic concept has become much more pronounced. And as I say, with good reason, because a lot of molecules made it into phase 1, but just couldn't make it out of phase 1 because of CMC issues. So now, at least, I think in a lot of companies, CMC folks have a voice at the table. So how do you assess these properties? So each functional area or functional group have their own criteria for how they're going to select. And again, this is not an exhaustive list. It's really just kind of high points. But, in medicinal chemistry, it's going to be typically the potency, the selectivity, any off-target, and often, obviously, intellectual property, which is an important component of selecting molecules. You want to have your IP in place. On the sort of metabolism side, you're looking at the ADME properties. You're looking at all the preclinical PK. You're trying to project what the human dose may look like, and also you're trying to correlate your PK/PD so that you have confidence in this molecule that it can hit the targets it needs to hit, that you can get the exposures you need to get, and that you can get the efficacy that you're expecting. On the toxicology side, there's obviously in this preclinical space, there's a lot of work done on the toxicology side to support your entry into the clinic. So you're going to do studies like dose range finding studies. You will do a GLP toxicology study of whatever the appropriate duration, anywhere from typically, let's say 28 days out to 3 months. You have to complete a safety pharmacology package. You need to understand any genotoxic implications of this molecule. And you need to define essentially what your clinical safety margins are going to be so that you can set out to design your phase 1 program and, within the bounds of the toxicology data or the parameters. On the CMC side, you do need to obviously look at the chemistry because, if you don't have good chemistry, you can't scale up. If you can't scale up, you can't actually run your preclinical program. You need to look at the formulation, both, as I said earlier, for your toxicology studies and also for your clinical studies. You need to understand the physiochemical properties. Typically, solubility is the one that really folks spend a lot of time trying to understand, especially for molecules that have poor solubility. Because it can give you some clues as to how you might want to formulate it or what types of formulations are necessary. And again, you need to understand the stability in terms of making sure that this molecule is stable enough to have a reasonable shelf life if it's successful. So this slide is really sort of speaking to, let's say, that transition where your program, let's say, is moving from the discovery world into the development world. And this is not just CMC focused in a sense. I'm speaking from a CMC perspective, but a lot of the functions have similar transitions. So, the transition from the drug metabolism group that's in discovery into development and so forth. So I just put this slide together because I think it's important to sort of understand that there is a difference in how folks view a program depending on whether they're on the discovery side of the line, let's say, or on the development side of the line. So Just a couple of points here to sort of outline that. I think in terms of expectations on a timeline horizon, at least from my own experience when I worked in discovery, you're waiting for the next data set, which is in sort of days, or these days, I guess, with so many contract research, maybe in the context of weeks. So you're expecting, assay results or whatever it might be to come within the next week, typically. On the CMC side, most things move at a almost geologic pace, I would say, compared to discovery, where very few things take less than a month, almost nothing, and most things are talked about in quarters. So you've gone from a mindset of days and weeks to get results to months and quarters, which can be quite a startling transition for folks who maybe have a discovery background and work on their first CMC development project and just can't understand why everything takes so long. And I guess that can be frustrating. In terms of planning, again, based on my own experience, the data that you're going to get is for your own function. So if you're a medicinal chemist, you're waiting for certain data to come in that's going to influence your next decision, and maybe not the decision of other functions. In terms of CMC development, everything has to be so coordinated, and it's driven sort of at a project level. So when I say coordinated, I mean, if you're responsible for delivering API, you need to know all the studies that everybody's going to do so that you can deliver API to not just the formulator, but to the pharmacologist, to the toxicologist, to whomever needs it. So you need to sort of understand all the moving parts, even though they're not directly within your function. In terms of the cost, I mean, discovery in a relative sense is a low cost set of activities. As soon as you move into development, the costs, they really do get high very quickly. So, when you're looking at, let's say, first campaign to make a GMP delivery of API, for example, costing $500,000, that's, quite expensive compared to the $25 you paid for your reagent, let's say, to make 20 mgs or 100 mgs or whatever it might be. The other thing is about regulation. So I guess, not to say there's no regulations in the discovery world, there are some for sure, but in a general sense, it's minimum. Whereas when you step into the development world, you're into a fully regulated environment. You're dealing obviously with agencies like the FDA or the EMA or wherever you're doing your studies. A lot of your activities fall under the context of GMP, which is a much more stringent, let's say, set of rules and regulations that you need to follow. So again, that can be quite a culture shock, because things that you can sort of do very quickly in your own lab as a discovery chemist, you really can't do that quickly. And it's also reflected in back here how long things may take. In terms of documentation, again, on the discovery side, you have a lot of study reports and things. They may or may not be audited or quality assurance stamped or whatever it might be. In terms of the development side of things, everything is going into your filings, so documents need to be reviewed, protocols need to be written, they need to be reviewed and approved. Quality assurance needs to sort of stamp all your documentation to make it acceptable for the filing. So again, it's a lot more in the detail. In terms of technical quality, again, when I worked in medchem, you would ask somebody what's the compound look like, and the typical answer is the NMR looks good, which is perfectly fine for and appropriate for what it's going to be used for. Again, moving into the development world, you have validated analytical methods. You need to quantitate your impurities. You need to understand the residual solvent levels. So it's a lot more detail expected, let's say, from materials that you're going to use. In terms of understanding other functions, I think the necessity for understanding development, I personally believe, is very important for folks who are in discovery, especially if they're going to be involved in projects. If you're working in a small company, especially these days where you're wearing, let's say, many hats and you're expected to maybe help move your programs into development, I think it's very important that you have some, reasonable understanding of what happens in development, because what I see the most is that folks can have some very unrealistic expectations and get very frustrated because, as I say, something that they feel should only take a week takes a month. Something that the quality looks fine to them, they realize, oh my gosh, I got to, spend 3 months having somebody validate all my methods. it's about having realistic, let's say, expectations of what development really means. On the flip side, for somebody who works in CMC, understanding how the mechanics of discovery, to be honest, is not necessarily that important. You don't choose the molecule. Essentially, you're presented with the molecule, and you're forward-looking in a sense that, what happened in discovery is fine. It got you to the point of selecting a candidate, but not so important for the future activities. So putting all of that into a sort of a graphic, I mean, the sort of activity, the types of activities that are happening that we kind of define as preclinical activities are, I would say, in three buckets. You have the pure discovery activities, where you're essentially involved in lead optimization. So, you have a reasonable scaffold, you have confidence that you're going to be able to select a molecule from what's happening there. In this center section, which is really all the preclinical development, you have some folks from the development world involved, and you have some discovery folks obviously involved, since they have all the history, all the information. So there's a sort of a informal, I would say, handover. And it depends on the company as to how that may happen. And then after you've filed your IND and you're in the clinic, it becomes a pure development project, to be honest with you. So in this section in the middle is really where things are overlapping. So depending on the organization, if we sort of look at the logistics of how these things happen, often there's a few potential candidates that are in play. And typically usually you want to get some toxicology information to give you an idea about how they may behave. And, you may have to make a few 100 grams. You most likely will need to select some form, whether that's the free form, salt, polymorph, or whatever it might be. And you'll need to develop a formulation for those DRF studies. Some companies run all of these activities in the discovery group. Others actually run all of these activities in the development group. And to be honest, there are some in the middle who kind of share the responsibility. Ultimately, whoever does it essentially, but you put it into your dose range finding study. And depending on those results, you may then at that point select a candidate or nominate a candidate. After you've nominated the candidate, different sets of activities need to take place. So you obviously need to develop a route to make the material. You have to manufacture the material, identify your phase. You're going to run your toxicology studies, make your clinical formulation, manufacture your clinical formulation, and then do all your stability work, and eventually put all of those data into your IND filing. Typically, in my experience, these activities in the past, some of them also happened in the discovery world, but more recently, a lot of these activities have sort of moved into the CMC, let's say, development world, because there's a lot of logistics and a lot of things, especially these days where, most companies, or not most, but a lot of companies are essentially virtual, have no access to their own internal lab. So all of these become contract, research companies that you need to deal with, and can be a lot of logistics. The timeline for all of these activities typically is between a year and a year and a half. Now, this can be a little bit debatable because, how do I say this? It depends when the clock starts. So different companies nominate a candidate based on different data sets and often measure the time only from candidate selection to, let's say, the start of the clinical studies, and use those as benchmarks. So it depends on your company and your company's sort of approach to this, but typically from sort of our experience of from late stage lead optimization into the clinic, it's usually more than a year. So what can influence the timeline? Obviously, the quality of the technical package from the discovery folks, and usually that means the synthetic route. If it's a relatively straightforward synthetic route, then you can scale up material a lot faster. If it's a very complicated route that requires many steps or reagents that are not really commercially available, then that can obviously take a lot longer. The other thing is, I mentioned that typically it's 1 to 3 candidates in play, but the amount of juggling, as I put it this way, can really influence the timeline, because I hate to say it, but how do you say it? Faraway fields are greener. So you get data on one compound, and it doesn't look exactly the way you want it, and you think, okay, the next one will be better, and it's harder to pull the trigger. So then you end up having 5, 6, 7, 8 compounds in play all at the same time, and it becomes actually difficult to make a decision as to what is the best compound. The other, as I mentioned earlier, is what are your criteria to select a candidate? So essentially, when does the clock start? And as I say, some companies we've worked with, candidate selections after their dose range finding study. Other companies don't make a selection until they finish their, let's say, 28-day toxicology study. So it depends on company philosophy as to how long these, not the individual activities, but the start to finish for this process, how long this all takes. It also depends very much on the risk posture for your organization. So you'll hear terms like phase appropriate and things like that. Each organization obviously needs to determine what they believe to be their version of phase appropriate. Obviously, the more conservative you are, the longer things will take because you'll want more detail and more information. So that can certainly affect the timeline. The other aspect is how much pre-investment are you willing to take on. So essentially, if we see a lot of companies who have set on the scaffold, for example, of the molecule, but don't quite know what the exact molecule will be. Go pre-invest, develop chemistry to make the scaffold, and have that happen in parallel to their compound selection, which saves time because once you select the compound, your synthesis will be shorter, for example. Again, and into that, it's a question of whether you run activities sequentially or in parallel. So the more risk you take, the more things you run in parallel, the faster your overall project will move. But then if you hit a roadblock, you've obviously spent a lot of money that maybe you can argue later with hindsight that was not necessary. And finally, the technical complexity, you can't overlook that. This can often be the biggest time sink. So as I said earlier, the synthesis, if it's a very complex synthesis, it can take a lot of time. The phase selection, so let's say your compound is amorphous and you want to have a crystalline form, this can also take a lot of time if the program demands that it be crystalline. The formulation, again, to come up with the right formulation for either your toxicology studies or the clinic, again, can sometimes be very technically complex. The issue with these is, the synthesis is usually obvious because you know what's already been done. For phase selection and formulation, the sort of more difficult part is to predict how long they'll take if they are more difficult than you expect, because it requires a lot of lab work to figure out how to solve some of these problems. So this is not meant to be read. I just wanted to put this in to say that a typical sort of timeline chart, if you want to call it that, for CMC in this space may look something like this. So the reason I just put this in, so I'm not expecting anybody to be able to read it, but is to just show that there's a lot of activities that need to happen and a lot of activities that are happening in parallel, irrespective of your risk posture, let's say. They'll be more or less compressed depending on how much risk you want to assume, but it requires a lot of coordination, a lot of different contract research organizations, a lot of different activities all happening at the same time, a lot of interdependent activities. So it can be quite a project to manage, let's say, you're in this space. After you've gone into the clinic, the project becomes much easier to manage because now it's about supply and resupply. But here, lots of things are on the critical path. So it can be quite complex. And as I said earlier, a lot of those logistics are interdependent. So you do need, the real key to success from a CMC point of view is actually planning. So you need a lot of planning, a lot of coordination. And the other thing is it needs active management. So just, let's say, finding a vendor to make your drug substance isn't really good enough. You need to follow up with that vendor. You need to make sure that they're doing the right thing, they're meeting your timeline and so forth. So it requires a lot of active management. So what kind of things delay? the preclinical work that are related to CMC. So obvious one is there's an interruption of the drug supply. So maybe you can make 10 grams, but now you can't scale up to make the kilo you need, let's say for your toxicology study, for example. I mentioned this earlier, but amorphous versus crystalline material can often be a difficult challenge to solve. One we see quite a lot of is that folks underestimate how difficult it may be to have a formulation for their toxicology studies, because essentially you need to hit multiples of your expected clinical dose. And sometimes that can be quite difficult, especially if your compound has very poor solubility. Related to that, there's all the complexity associated with different classifications of molecules. So whether it's class 2 or class 4, meaning it has low solubility and it can also have low permeability, which is a very difficult situation to be in. And the need for an enabled formulation, essentially meaning that you need a much more complex formulation than simply putting your powder in a capsule or even making a tablet, that you need to essentially manipulate the physical property of the drug to enable it to be formulated. Again, stability issues do occur, and they're not generally predictable. Most often they're in the drug product, and most often they're associated with materials that are amorphous. So amorphous materials typically have poorer chemical stability and also poorer physical stability than crystalline materials. And there's always the bad day, let's say, when a new polymorph or a new impurity shows up that you haven't seen before, and you obviously need to take time to figure out how to fix or solve that problem. And finally, I've mentioned this a few times, but I would say you can't underestimate the logistics of engaging a particular CRO. When I started consulting first, I was horrified to figure out that in some cases it can take months, just to get a piece of paper signed, get a confidentiality agreement signed before you can even tell the CRO what you want them to do. Sometimes these activities can take months, quarters even. So, again, don't underestimate how long it can take to get some of these things, let's say business-related activities in place. So I guess to kind of finish up, I mean, the CMC activities in this space are pretty much all on the critical path. In essentially, the drug substance is usually the one that's most critical, because without it, you can't run the program. Without it, none of the other functional groups can start any of their activities. You can't start the toxicology. So all of that's related to drug supply. And at the beginning of these programs, obviously the drug supply is the most critical. But pretty quickly, some of the other activities do go on the critical path. So essentially, the way I would describe it is most of the CMC activities are actually on the critical path to get from candidate selection into the clinic. So when the folks engage the CMC folks, I guess, at least in our experience, I'll be honest, most folks engage CMC, to be honest, only when they have a problem. Or after candidate selection, when sort of the logistical aspect of the project becomes clear and you realize, maybe I need some help, because there's just so many things to figure out. In our experience, I would say the optimal time to engage the CMC folks is towards the end, around the time you're going to select your candidate, because there you can have the most impact on the timeline and get the best understanding of what issues may be coming up during the preclinical workup, let's say, for the IND. So that's pretty much my talk on the CMC. Just 30 seconds to introduce Exemplify Biopharma. So we're a partner research organization. We have two complementary business units, I guess. We do full CMC consulting, and we also have a CMC laboratory. So we do process development, analytical and development, and early formulation development. And we have a number of clients where we pretty much run their CMC program for them in this space. And we have all of the different deliverables we can provide, and all essentially under one roof. We were founded in 2015, and we're located in Cranbury in New Jersey. And I guess our philosophy is that we like to work in a trust sort of environment, essentially. So we believe that in those types of environments, everybody does better. So we like to think that if you invested $1 with Exemplify, we could deliver you maybe $1.05 back in terms of value to your program, and also to help with your knowledge of institutional knowledge and provide all that information. In terms of how we operate, as I say, we have a consulting group, and we're doing more on the strategy and project management side, also including things like writing IND chapters for Module 3, providing quality audits and things like that. And then on the lab side, as I mentioned, we do process development, the normal things you would expect to see from a CMC laboratory, everything to support moving a program into phase 1 from candidate selection. So I'll stop there and thank everybody for your attention.
Wonderful. Thank you so much, Dr. Paul, for your insightful presentation. And as mentioned, if you want to connect with him and his team, Sandy and Kelly, that are also participating at the program, you can book a one-to-one meeting with them, and we can schedule that. And if there's any questions for Dr. Paul, you can email us. He is available via email introductions as well. And with that said, I do want to thank you again, Dr. Paul.
No problem. It's my pleasure. Thank you.
You're welcome. And with that said, we will be heading to our final presentation before our panel discussion, which will be Dr. Krishna. So just give me one moment.